UV excitable fluoroprobes have an inherent signal contamination because of the autofluorescence, mostly from NADH, limiting the use of fluoroprobe. Our method can be used to precisely correct the autofluorescence from NADH and to extract a true uncontaminated fluoroprobe signal. Demonstrating the procedure will be Jeong, a research assistant professor from my laboratory.
After allowing the myocytes to settle to the bottom of the tube, aspirate the supernatant. Label the cells with 2 milliliters of freshly prepared 1 millimolar fura 2 FFAM diluting solution for 60 minutes at 4 degrees Celsius. At the end of the incubation, place the tube in a 37 degrees Celsius water bath for 30 minutes in the upright position before removing the supernatant. Then add 4 milliliters of room temperature culture medium for storage at room temperature. For in situ isosbestic fura 2 FF point identification, mount the diluted cells onto the microscope stage and wait 3 minutes for the cells to sink to the bottom. Perfuse NADH-free 37 degrees Celsius calcium-free solution into the cell culture and locate a target cell. Perfuse saponin solution for 60 seconds before reperfusing with the NADH-free calcium-free solution.